thank you, and welcome everyone up to this SPP webinar. We certainly appreciate your interest in SPP and are happy to have this opportunity to help you guys learn more about who we are and what we do. We're also excited to be a part of dkNET and to working with them in the longer term to integrate SPP analysis into their resource. For now, though, we will focus on introduction to SPP itself, and with that, I will pass the presentation over to my colleague, Scott Oxner.
All right, thanks, Neil. And again, welcome everyone to the webinar on Signaling Pathways Project, or SPP. So we're going to start by defining what we understand as signaling pathways. The term pathways refers to a series of functional and physical interactions that constitute the cellular response to a signal. Now, this signal may be a hormone or some other signaling molecule. It may be an environmental substance or an event in the extracellular matrix itself. But really, any event at the cell surface or inside the cell that requires the cell to respond to it will modulate one or more signaling pathways. And now, cells respond to signals in many different ways, but arguably the most important of these are those that occur in the nucleus, and specifically those that result in regulation of gene expression. Individual elements, that make up these pathways, we refer to at SPP as nodes. Now, the three major signaling nodes encompass receptors, which initiate the signal, enzymes, which amplify the signal and modulate the activity of the third major flavor of signaling node, the transcription factors. Now, you should be aware also that there are some nodes, such as nuclear receptors, which integrate all three of these functions into one molecule. I should also mention here that there's a fourth class of SPP signaling nodes, the conodes, which is made up of molecules that don't easily fit into any of the other signaling nodes that I've already referred to. All right, so as those of you who are active in the research labs will be aware that most of our knowledge of signaling pathways is obtained in the research literature in the form of research articles or in reviews which aggregate facts from multiple different research articles. However, there's actually another universe of information that informs upon cellular signaling pathways in the form of archived Omics datasets. And again, for the purposes of SPP, we're going to focus on two specific types of omics, and those are transcriptomics, represented by expression array and RNA-seq, and citromics, represented by ChIP-seq experiments. So, what are these datasets and what information do they contain, and how can they help us understand cellular signaling? So looking at a basic representation of the cell, for any given cell perturbation, whether that's an siRNA experiment, a small molecule, transcriptomic experiments survey over thousands of messenger RNAs in a cell, and they tell you which ones are increasing or decreasing in abundance in response to that perturbation. Citromic datasets or ChIP-seq experiments, on the other hand, inform upon interactions between proteins and DNA. And so although they will not give information on which mRNAs are up and down, what they do show is where in the nucleus certain nodes interact with DNA. And in doing so, they afford us some insight into the underlying mechanism. But what's important to remember is that a specific signal can have multiple mechanisms of action. And so by combining the transcriptomic with the citromic experiments in a single resource, SPP enables researchers to focus in on specific mechanisms that might be mediating a specific signal. Now, even though omics platforms inform upon thousands of molecular events in a cell at any one experiment, as a consequence, of the way we do research, what often happens is that in order to get a paper published, the investigators have to focus down on a small handful of those events, and they have to look at these in more detail. But what happens is that there is all this other information that will not make its way into the article. Now, even though that information wasn't of immediate interest to the original investigator, it might still be of interest to other researchers in related fields. So, unlike research articles, which are presented in this example on the left very neatly and have large numbers of editorial staff working on them to ensure that they are clearly presented, the archiving of omics datasets is for the most part done in a way that throws up obstacles to their reuse. They're often available only in a raw form, as shown in this example, and very often they even lack the most basic information, such as the research article from which they were associated. Now, we are all, I'm sure we all have rooms in our house that look like this. I know I do. And I feel it's very apt analogy here. I see research literature as the closet in which things are neatly folded. It's very easy to find what you want and what you want to wear. However, on the other hand, if you don't take the time to do the actual folding and stacking, it's going to be very difficult to find what you want and when you want it, and that's exactly what you see with omics data archives. Now, some 15 years ago, as part of the Nuclear Receptor Signaling Atlas, or NRSA, we recognized the long-term value of transcriptomics experiments, and we decided to start organizing these data sets relevant to nuclear receptor signaling. Our idea at the time was to build a resource where researchers could retrieve information from transcriptomic data sets as easily as they could from research literature. Initially we focused on organizing the data sets themselves, and you may actually have heard a lot recently about the FAIR data movement and the increasing mandate from NIH to share the results of research. And as far back as 2006, NRSA was sharing data sets through its website and applying ideas such as DOIs for data sets, adding detailed metadata that have now since become cornerstones of the FAIR doctrine. And actually, in fact, in 2005, we were the first repository in the US to receive a DOI prefix from Crossref. So, this is all to say that really this is an area in which we have spent a long time thinking about. So a natural progression from organizing data sets themselves was to enable researchers to go further using a inquiry tool that we call Transcriptomic. And if you actually read the SPP article, you will probably have seen this slide here. Now, nuclear receptors shown here are, of course, only one very small sector of a large number of nodes that make up cellular signaling pathways. So in order to build a resource with broader value for the cellular signaling community, we had to start thinking kind of outside of the nuclear receptor box. So what we've done is we've spent the last 2 years or so building the successor to the initial nuclear receptor-centric resource that we refer to as the Signaling Pathways Project, or SPP. For transcriptomic expression array and RNA-seq experiments, we, 1, defined what node was manipulated, and 2, how it was manipulated. Was it treatment, for example, with a small molecule? Was it loss of function through a knockout? Maybe a gain of function through an overexpression? For ChIP-seq experiments, we asked what was the node antigen that was used in that experiment. And for both experiment types, we also logged what cell line the experiment was done in, as well as the species. As a result, we have now two universe of omics data points, one for transcriptomic and one for citromic, that we have mapped to cellular signaling nodes that they were initially designed to inform upon. Now, the key to all this is that we use a consistent set of terms to describe various facets of the experiment, so that when it came to designing our user interface, it would be easy for users to mine seamlessly across the entire data matrix. So here we're going to switch. Actually, building the SPP database was only part of the work. In order to make all this curated data accessible and reusable by researchers, we had to build an intuitive user interface that did not assume any specific level of informatics expertise on the part of the user. So users can actually use SPP in two ways. They can browse and they can search. So first, I'll briefly discuss browsing. Let's say a user wanted to browse or search for all of the data sets relevant to a particular node family. Now they can do so in the datasets section, where they can use the drop-down menus to define their nodes of interest. So let's say I'm interested in, I don't know, ChIP-seq experiments relevant to the CBP-P300 family. The first thing I do is I enter enzymes, then acetyltransferases, and then CBP-P300. And what you get is a filtered list of just those experiments in SPP dealing with those specific filters. Now, one thing to note is that these menus represent the theoretical scope of SPP. And for a variety of reasons, either the data sets don't exist, two, they haven't been archived, or three, we may not have curated them yet. Not all of the node families that you see in these drop-downs will be represented in SPP at any given time. All right, so now one of the aims of SPP and of dkNET is to make it easier for users of these data sets to cite them, so that the original investigators receive attribution when their data sets are reused. And in order to assist in this process, we've gone to a lot of trouble to make it very easy for researchers to download and add the citation to their favorite citation manager right here. We also always provide the link to at least the Medline abstract of the original article with which the dataset was associated. And as I showed you earlier, this is often absent from the original public archived records. And so figuring out what and where the original associated article is involves a lot of effort, but where possible, we fill this gap in annotation. Now, browsing of data sets is fine, if you're just looking for a specific data set. But what if you want to ask questions over the wider universe of SPP data points? So in order to do that, you would use OMINER search engine. Now, what I'd like to show you now are some examples of things that you can do with in OMINER to gather evidence for regulatory relationships between signaling pathway nodes and downstream transcriptional targets. So as I go through these examples, I want you guys to focus on the relationship between how we define the hierarchy of nodes in SPP and how data points are organized in the user interface itself. So for all three nodes, and here we're using receptors as an example, there are three levels. There's the category, the class, and the family. And that hierarchy is reflected in what we refer to here as the regulation report. All right, so I'll start with a use case that concerns a lipid transporter whose decreased expression was recently linked to defective insulin secretion. And here we're going to use SPP to determine if regulation of STARD10 might help us hypothesize a mechanism for known links between cellular signaling nodes and insulin secretion. So let's go to O minor, and to do a single gene search, we're going to use our drop-down and select single gene. We're going to select our omics category as transcriptomics, and we're going to begin to enter a couple of key letters of our gene that we're interested in, STARD10, and we're going to select that from our exact matches. And for purposes of this example, we're just going to keep everything at default, but know that all the results were at least a p-value of 0.05 or less. So we click the submit button, and what we see are a number of data points for STARD10 that are downregulated here in blue or are upregulated depicted here in red. And of course, you see all your categories. You see receptors, enzymes, transcription factors, as well as conodes. If you actually click on a data point, you get a wealth of information about that particular experiment. Of course, you see the gene that's being interrogated. You see it was twofold upregulated. You see a p-value significance for that fold change, as well as a description of the biosample that was used, an experiment name, and here we're using a TLR1/2 ligand compared to a vehicle. So let's say, for example, you don't know what P3CKS4 is. We have links here to a bioactive small molecule, and what this does is gives you a link directly to PubChem. So you can find out exactly what P3CKS4 is if you're not already familiar with that molecule. And you can see from its pharmacology that it is a TLR2 and TLR1 receptor agonist. Additionally, what you can see from these data points, down here on the More Information button, is the experimental and dataset information for that fold change. So you get the experimental name, which is the treatment versus vehicle here, a description of what that was actually done, the biosample, the species, and you can see what data set that experiment was a part of. Now, also, if you want to go directly and look at that data set to maybe see what other experiments were done, we now have a hyperlinked DOI that if you click it, takes you directly to the data set page, and all of the various experiments that were done in that data set. The one we were looking at happened to be experiment 2. All right. Let's go back to our page here. So so that was showing you evidence of the transcriptomic regulation of STARD10. And what I'd like to point out is here we see some evidence for nuclear receptor regulation by estrogen receptor. And here we see 2 blue data points, or in other words, downregulated. And if we click on our information, we happen to see that these are ER alpha knockdown experiments. We also see some experiments treated with 17 beta E2 and ER alpha ligand. And we can see that treated with ligand, these an upregulation of STARD10 in this specific cell line. So what we see is we definitely have some evidence of estrogen alpha regulation of STARD10. So now let's take a look at the citromics data set. So we recommend that when you switch searches like this, that you reset your filter. And so we're simply going to select single gene, but now we're going to filter on citromics only. Again, we put in a couple of keywords. We select our gene of interest here. Again, we're going to go default. We click submit. And what we see here now are a number of data points that show protein binding in the STARD10 promoter. So each one of these data points is a ChIP-seq data point for a particular protein, what I want you to notice are all these ER alpha data points in combination with various ligands and other proteins. But you can definitely see there's a lot of good evidence that ER alpha is binding the STAR-D10 promoter. Okay, so just looking at SPP alone, there is an abundant evidence that the estrogen receptor regulates STAR-D10. However, we could not find any mechanistic literature evidence, in PubMed for the estrogen receptor transcriptional regulation of STAR-D10. But what is known from the literature, however, is that pancreatic insulin content is actually regulated by the estrogen receptor. So if you take all of this information in total, and if you visualize STAR-D10, the estrogen receptor, and insulin as three corners of a triangle, now the literature makes some connections, but SPP forms the third side of that triangle and allows the users to hypothesize a possible mechanistic basis for the ER insulin connection. And that hypothesis may that estrogen receptor is regulating insulin status in part due to its transcriptomic regulation of STAR-D10. All right, so sometimes these hypotheses can be difficult to generate if you only have a specific gene in mind. And so for this reason, we allow users to back their queries out, sort of zoom out, and encompass a broader range of cellular functions using Go terms. An example of hypothesis generation, again, of relevance to NIDDK, I'm going to pose here as an investigator seeking novel mechanistic evidence underlying the known link between EGF and brown adipose tissue formation. All right, so I'm going to go back to our live demonstration here, reset our filter, and we're going to enter in a Go term search, and here we're going to begin entering a couple of keywords to bring up brown fat cell differentiation. We'll select that. Now this serves as a proxy for a number of genes involved in brown cell fat cell differentiation. But know that on a run of a Go search, you need to specify the node class. And here we're going to select receptors. And this is to prevent potentially very large queries large queries being submitted that might impact server performance. And so I've selected receptors and catalytic receptors. I'm going to click submit. And the default data display is the target display, meaning that these are all of the gene targets that were in the Go ontology term for brown adipose tissue formation that are also have evidence in SPP. Now, in order to narrow these down, what I can first do is I can select the category view. And what this does is now it breaks down those same data points into receptor families that may be involved. And so since I'm specifically interested in EGF receptors, I'm going to go back to our form, and I'm going to narrow down my search just to the EGF receptor family here and click submit. And so what this does is now I'm only looking at those brown fat cell data point genes, related genes that have something to do with EGF receptor, either down or upregulated in blue and red. And in the regulation report, I can see evidence for EGF receptor family regulation of PPARG gamma. Now, this is actually consistent with what is known in the literature, that PPARG gamma is regulated by EGF receptor family members. And so now this is actually kind of a positive control for my search, which gives me more confidence that the results I'm getting back from SPP are real, and that I can believe the other evidence that I see. So really what we really want to know is what are the novel EGF receptor family targets? And so what I want you to notice is that two different EGF receptor family ligands, TGF-alpha and NRG1, both of these repress laminin-4 expression. Now, this is interesting in the context of evidence from the literature suggesting that laminin-4 is a negative regulator of brown fat tissue, of fat browning rather in mice. And so again, we have this complementary relationship between the literature and SPP, whereby the literature provides the outlines of the hypothesis and SPP provides evidence for the mechanistic basis of that hypothesis. Now, another way you can query SPP is by using your own gene set. So again, to illustrate this with an example of relevant to NIDDK, what I'm going to do is I'm going to pose as a researcher who's looking for novel receptor signaling paradigms in Crohn's disease. And as for my research, I'm going to take a set of genes that was identified in this paper to have prognostic value in Crohn's disease. So we'll go back to our search. We're going to reset it, and here we're going to select the option of gene list. We're going to click choose file so that we can select our Crohn's signature. Now, this is a comma-separated file of those genes implicated in Crohn's, their official gene symbols. So now I've uploaded my gene list, and I'm going to focus down here on catalytic receptors. Hit submit. And what I see is a lot of evidence connecting various catalytic receptor signaling systems to transcriptional regulation of those genes implicated in Crohn's. So as a researcher in Crohn's, I already know that there's a documented relationship between HDF and Crohn's disease. So I'm going to explore that by narrowing down my search to just the HDF receptor family. And again, it's always nice to have positive controls, and here's one. So there's evidence that CXCR4 here is repressed by an inhibitor of the HDF receptor, right? And now this is consistent with what has been previously shown in the literature, and that is HDF regulates CXCR4, right? So now, what you also see, there are numerous other genes here that SPP provides evidence that are transcriptional targets of HDF. And one of these is the chemokine CXCL11, for which there is a large number of data points showing repression of CXCL11 in numerous independent experiments from actually the same inhibitor of HDF. Again, however, we were able to find no evidence for this regulatory relationship in the literature. So when we combine this with SPP regulatory evidence with the literature evidence that CXCL11 is consistently overexpressed in Crohn's, we've closed that triangle again on our hypothesis that HDF is connected to Crohn's, at least in part, through the induction of CXCL11. Now, something else that we introduced in our recent paper was the concept of consensomes. And what consensome analysis does is it surveys over all of the experiments that map to a specific node family. Using the example here, we're going to use insulin receptor. So we'll select consensomes, we'll select receptors, catalytic receptors, and the family of insulin receptor. And we click submit, and we see our consensome report. And Using the example here of this insulin receptor is to rank the genes by strength of their regulatory relationship with nodes in that family. So what you're looking at here on the left are the list of genes that are consistently regulated in insulin receptor interrogated experiments. You see a discovery rate. Now this is the frequency with which that gene is differentially regulated in those specific experiments. You also have a geometric mean fold change, giving you an idea of the magnitude up or down. And you also have a consensome p-value, gives you a sense of significance that the result you see is not due to chance alone. And then we rank these genes into percentiles. So, you know, a representation that isn't available on the site right now, but one that I think makes this idea easier to understand is plotting these points in a scatter plot, where on the x-axis you have the discovery rate, which again is essentially the frequency over all of the experiments in which that specific messenger RNA was significantly differentially expressed. And on the y-axis here you have the rank of that gene in the consensome itself. So typically up here at the top, you've got genes that are highly responsive to the insulin receptor, less so at the lower discovery rates here. And at the bottom, you have those genes that are very weakly or are unresponsive to insulin receptor signaling. Now, just as a technical note here, you're going to notice this interesting two-tailed distribution that we see in many of the consensome scatter plots, where genes of the same discovery rate actually have different consensome p-values and therefore different rankings. The reason for this is that genes in the upper tail were assayed in larger numbers of experiments than those in the lower tail. Therefore, they're assigned a lower p-value due to the increased sample size. So when you look at consensomes, you know, this is one caveat to be aware of. Now, so by ranking genes based on their responsiveness to a specific node, consensomes offer us a unique environment where the user can actually use SPP to formulate new hypotheses that they wouldn't have been able to using only the information in research literature. So what we've actually found, having looked now at consensomes from many different nodes, and again, using the human insulin receptor here as an example, is that the most highly ranked genes, in addition to those containing those that are known targets of the insulin receptor, we also see many genes that have no evidence of being involved in insulin receptor signaling. And so information like this can help researchers hypothesize novel mechanisms of action and signaling systems that have not been explored in the research literature. So to exemplify that concept, we're going to look at the relationship between insulin and autophagy. So what is autophagy? So autophagy is the process by which cells, in response to depletion of their extracellular nutrient sources, attempt to maintain energy levels through metabolism of nutrients within the cell itself. And this process is actually well known to be regulated by insulin, which is, of course, the primary signal of the fed state. Now, insulin regulation of autophagy has been very well documented in the research literature at the protein level through regulation of AMPK. Now, interestingly, and this is something that has not been explored in the research literature, key factors that are involved in the regulation of autophagy are actually highly ranked in the insulin receptor family consensome, indicating that they are transcriptionally very responsive to signaling through insulin receptor family. And these include, for example, ULK1 here, ATG14, also known as ATG14L. And in fact, 33 of the autophagy pathway genes, of the 33 autophagy pathway genes in the human genome, nearly 50% of those are in the 90th percentile of the human insulin receptor consensome. So this leads us to the hypothesis that in addition to regulation of autophagy at the protein level, signaling through insulin receptors supplements that level of regulation by modulating the expression of messenger RNAs and coding factors in the autophagy pathway. All right. So now here, if you're looking at the regulation report for the most, well, here, first let's filter on HEG14, one of those autophagy genes that was highly implicated in insulin receptor signaling. Now, if we click on that gene, it takes us to a single gene transcriptomic report for the most highly ranked factors. And remember, it maintains our filters that we initially picked for our consensome. So what we're looking at is a single gene search of AGT14 for catalytic receptors, insulin receptor family. But what's very clear is that insulin signaling exerts a profound negative effect on that gene's expression. Multiple different insulin receptor family member agonists all repress expression of HEG14. But what is equally, if not more striking, is that over half of those data points, the ones you see here in gray, those are far below the two-fold cutoff that is typically widely used by investigators when they evaluate results of an expression array or RNA-seq experiment. So if an investigator was looking at this experiment right here, for example, in isolation, he'd not be very convinced. The fold change is 1.3, even though that p-value is significant, still it's not a very big fold change. However, if you take the sum of all the data points which you can visualize in SPP, now a very clear and consistent pattern emerges, and that is the autophagy gene HEG14 is downregulated by insulin receptor signaling. So our running theme, throughout this presentation has been the generation of hypotheses, and to make the hypothesis generation power of SPP available to as many researchers as possible, we're involved in a number of different collaborations. The most important of these is our collaboration with our sponsors and today's hosts, dkNET, and the goal of which is to build a hypothesis center that will then help dkNET users formulate researchers' hypotheses connecting cellular signaling nodes to cellular processes and normal human health and disease states. In addition to our work within SPP, we're also exploring other ways to leverage the work we are doing here at SPP. We're in discussions with PLOS to launch a new collections on their site that will enable PLOS readers to link from articles to the formatted data sets on the SPP site, giving them access to a wealth of information that would not usually be available through, for example, a direct geo search. We're also working with the IDECR group at UCSD to make SPP regulation reports available as networks through index pathway exchange, and this collaboration is via the SPP API, which we would be very happy to discuss more with anyone interested offline. And finally, with the support of the American Thyroid, I forgot one here, we're also working with the Reactome Pathway Database to complement their literature annotations with our own curated data points. Finally, with the support of the American Thyroid Association, we curated a collection of data sets of relevance to researchers in the thyroid receptor signaling community. And so, as I said back at the start, there's a lot of very useful, detailed information on cell signaling in research literature. But there's also a lot to learn about cellular signaling that is not described in that literature. And what we at SPP are aiming to do is to help researchers fill those spaces so that the field as a whole can advance to a broader understanding of how cells respond to signal. All right, does anybody have any questions?
So if you have question, you can unmute yourself. The button is on the left lower side. So are there any questions? Let me check the chat. So there is one question. Does SPP cover all disease field? Like what kind of disease field it's focused on? And if, let me see what is it. If I found some useful data set, can I deposit or suggest a data set to SPP project?
I can take the second part of that question. Yeah, if you have a data set that you want to be included in SPP, or if you found a data set you might think is interesting, by all means. What we first recommend you do is to deposit that data set, if you can, if you have ownership of it, into a public repository like Geo or ArrayExpress, so that it can benefit the community as a whole, but also it will facilitate us grabbing that data quickly and easily to import it into SPP. Now, I'll hand it over to Neil to answer the question about what disease scope SPP covers.
So most of our effort is focused on the annotation of data sets that involve manipulation of signaling nodes. However, we are also in the future we are going to release a number of experiments, number of data sets which are of relevance to NIDDK, such as diabetes, and so we will be releasing those in the future, so that when you run a particular search for a factor of interest, you will see experimental data points from those data sets side by side with data points from experiments involving the manipulation of signaling nodes.
Okay. There is another question. Maybe it's a comment in the chat room from Cherry Watson. Is how incredible powerful from the last bit years of arguing with reviewers about whether small but significant and reliable changes are important could be assisted by this.
Well, so thank you for that feedback.
So are there any more questions?
Great presentation, thank you.
Thank you.
Okay. If you still have more questions, you can send Neil and Scott emails. Okay. So thanks for Scott and Neil for the great talks, and thanks for everybody joining today's webinar. The recording should be available next week on dkNET YouTube channels. We'd like to encourage you to share today's webinar with your colleagues and students. To receive upcoming webinar information, please subscribe to our newsletter or check announcement at dkNET.org. Thank you again and have a great weekend.
Thank you, everybody.
Thank you.